I hope you've had an outstanding day full of good research discussion and networking. We started today with a really thought-provoking talk that highlighted the need and the necessity for innovation. And since then, you've hopefully seen a lot of great research, maybe checked out some of the posters on some of the projects that we've funded that hopefully will contribute to that innovation. We'd like to end today by sharing the story of some truly innovative research that has had and continues to have real impact on many lives. When the idea for the Dairy Innovation Hub was first developed, everyone had a strong belief that this should focus on more than just one single aspect of dairy. It should cover everything from proper use and stewarding of our land and water resources, to animal welfare and health, to human health, and to growing our farm businesses and communities. Furthering developing Wisconsin's safe and nutritious, abundant dairy products is good for the state, good for farms, and good for all of our nutrition. With that said, I'd like to introduce someone who has made a career of using dairy ingredients to help people. Denise Ney is a professor and registered dietitian, nutritionist in the Department of Nutritional Sciences at UW-Madison. She was also the director of the didactic program in dietetics for more than 20 years. Her research program addresses gastrointestinal physiology with a special interest in the dairy protein produced during cheese making, GMP, which she'll tell you plenty more about than I ever could. Ney pioneered the use of medical foods made with GMP for the dietary management of PKU, a rare genetic disease. Her work in this area even garnered her the title of Rare Disease Hero from the FDA. And now her work with dairy is also helping a new population, postmenopausal women. So please join me in welcoming Dr. Ney.
Thank you for that kind introduction, Heather. Can you hear me okay in the back? Okay. And thank you for sticking around to the very end. Our research with glycomacropeptide, or GMP, provides a prototype for how...
I don't think your mic's on. Or you need to speak like a bit closer. Okay. Thank you.
It says it's on. Is this good? Okay. Have to have it right up here. Okay. Our research with glycomacropeptide, or GMP, provides a prototype for how personalized nutrition with dairy changes lives. I'm going to start by telling you the story of how we developed GMP medical foods for those people with phenylketonuria, or PKU, a rare genetic disease. We'll move on to our mouse studies, which eventually led to the current GMP hunger study, a short-term high-impact study funded by the DIH. The GMP story begins with Professor of Food Science Mark Etzel, who developed a way to isolate GMP from cheese whey with high purity. GMP is a polarized molecule with a pKa of less than about 3.8, and Mark used ion exchange chromatography. Here you see the Bruker column that was used for our inpatient metabolic study. Mark is with us today. Mark? In my lab, we went on to conduct proof of concept studies in mice, and I eventually was awarded grants from the NIH and the FDA to conduct phase 1 and phase 2 clinical studies in subjects with PKU, many of whom came from the Biochemical Genetics Clinic at the Weizmann Center here on campus. This research, in conjunction with commercialization of GMP medical foods, has improved nutrition, and I would argue also quality of life, for thousands of people with PKU throughout the world. PKU is an autosomal recessive disorder characterized by a deficiency in the conversion of the essential amino acid phenylalanine to tyrosine. It is managed with a lifelong low alanine diet. This diet is essential to prevent brain damage. Hence, the warning on the diet soda cans that contain aspartame, a dipeptide of aspartic acid and Phe. The tragedy of severe cognitive impairment, the phenotype of untreated PKU, has been largely eliminated in the developed world due to newborn screening and the initiation of a low-Phe diet in the first few weeks of life. Newborn screening was invented for PKU. It was discovered by Robert Guthrie, and he developed what's referred to as the Guthrie test, which is essentially a bacterial inhibition assay, which is used or was used with blood drops in the newborn screening. Now, and he did this in 1962. Kennedys were in the White House. There was a big interest in mental retardation, as it was referred to at that time. On our campus, Harry Weisman was a leader at the national level to secure legislation requiring newborn screening throughout the United States, which was achieved by the late 60s. Newborn screening today is a lot more advanced, and Wisconsin continues to be a leader in newborn screening. The mandatory uniform screening panel requires screening for 30 disorders based on a blood sample. We screen for 47 disorders in a blood sample, and also hearing impairment and congenital heart disease. This is done with tandem mass spectrometry at the Wisconsin State Newborn Lab of Hygiene over there in Henry Mall. It's a wonderful public health program. With respect to PKU, we have about 5 to 8 babies born each year in Wisconsin diagnosed with PKU. PKU is caused by over 800 mutations in the hepatic phenylalanine hydroxylase gene, which has as an essential cofactor tetrahydrobiopterin. When a baby is born, there's nothing that you could tell by looking at them or examining them that they have PKU, okay? But within 24 hours of consuming breast milk or infant formula, a normal high-protein substrate, they will have blood Phe levels 10 to 20 times normal. And of course, that would be picked up on the heel stick, the blood spot, which would be used for newborn screening. Many of you probably remember that test that your children had before they left the hospital. Okay. With PKU, we see very high blood Phe levels. That translates into high brain levels of Phe, which impairs brain development by poorly understood mechanisms, multiple mechanisms likely. The cornerstone for management of PKU is the low Phe diet, and it has two components. The first is that all protein foods are eliminated. So what can you eat? You can eat measured amounts of fruits and vegetables, and none of the high protein vegetables like beans or nuts or anything like that. Then, for nutritional adequacy, a medical food is required to provide sufficient calories, nitrogen, vitamins, and minerals. Now, when we came into this work in 2003, there was only one type of medical food available for management of PKU, and that consisted of amino acid formula, that is mixtures of amino acids. You leave out the phenylalanine, and most people would have to drink about a quart of this amino acid formula every day for their diet, along with a small amount of fruits and vegetables. The amino acid formula is best described as smells bad and tastes worse. The food scientists never could do much with those sulfur amino acids to improve it. Fortunately, adherence with this amino acid formula is pretty good in infancy, but as you can imagine, once the kids, especially when they start school and then on through life, the compliance is really very poor with this highly restrictive diet, and the stakes are very, very high, okay, in terms of the effects on the brain, you know, in teenagers, throughout the lifespan for someone with PKU. All right, let's talk about GMP a little bit. This is a 64 amino acid bioactive peptide. It occurs in milk within the kappa casein micelle and is released during cheese making, where it is isolated, or it can be isolated. Now, GMP is a very unique protein. It has a very interesting, not necessarily positive, but I'm going to call it an interesting amino acid profile. And it is the only known natural dietary protein that does not contain Phe. Hence, its application to PKU, which Mark Etzel and I recognized very early. The GMP peptide is also enriched in threonine, in branched-chain amino acids, and has a lot of glycosylation with mucin-type carbohydrate chains. Many of those carbohydrates include a lot of sialic acid, which is enriched in breast milk. Now, these mucin-type carbohydrate chains on the GMP peptide, they confer prebiotic properties on GMP. So now, we heard this morning very nice talk from you in Brad Bowling's laboratory about yogurt, probiotic, put the organism itself in the food. I'm not talking about that. I'm talking about a prebiotic. I'm talking about carbohydrates which are poorly digested by the mammalian gut, but are fermented very nicely by healthy bacteria in our own gut, which provides a very diverse, healthy microbiota, okay? You're giving the bacteria the food they like to eat, okay? So I point this out because to have a protein source that is a prebiotic is very unusual, okay? Most prebiotics are essentially carbohydrate-rich foods, including onion, garlic, asparagus, my personal favorite, the banana, okay? But here we have a protein that is a prebiotic, and I believe this underlies many of the positive effects of GMP, which I'll tell you about. Here we see Jessica, and she is 10 years old, and she is sampling pudding, in particular, low-Phe GMP pudding for the first time. Jessica is currently a successful college graduate living on the West Coast. A lot of time has passed, Mark. All of these foods that you see here were designed by Kathy Nelson in the Center for Dairy Research here on campus. And I want to emphasize this because this was a crucial contribution to move this research forward. So, to tell you a little story, you know, we all need money to do research, okay? Nothing goes anywhere without money. And so when I started in this was like a side project, okay? I didn't put a student on it or anything because I didn't know where I was going to go. I was able to develop a PKU mouse colony because I got a TIF grant, a TIF Draper grant. That's WARF money that comes through the graduate school. So I had about $100,000 to get going. I also had people from the PKU community phoning me up and said, I heard about what you're doing. You know, we have a foundation. I want to donate money. The Michaud Foundation came through, you know, to donate some money just to support this research. So I got through the mice. It looked good. I had the proof of concept that I needed to show that I could make a complete protein out of the GMP and that it would support the growth of a weanling mouse. That's your gold standard in the animal field for development. And then I found an RFA from NIH. And that RFA was for unique studies for obesity and metabolic disease. So I thought, okay, I can fall into metabolism. And I called up the program officer. I described what I had, and I had the mice, and I wanted to develop GMP medical foods for humans with PKU. And she said, oh, this is very interesting, but don't send us the grant until you can demonstrate that you can make foods out of this GMP and that people with PKU will eat those foods. Now for me, as a nutritional scientist, this is a little bit out of my scope of practice, development of foods. But I went to the CDR and they assigned me Kathy Nelson, who was a very experienced food scientist, had a career at Pillsbury, I think, in Minneapolis. And she was only working part-time, but they put her on the project. They said, sure, that's part of our mission to develop new markets for dairy foods. And she developed the foods. I conducted sensory studies to evaluate those foods and with subjects with PKU from the Weizmann Center. The grant went in. I got it the first time. So that enabled me to do a phase 1 metabolic study where I admitted 11 PKU subjects to the clinical research unit at University Hospital for 6 days. Because I had the NIH grant, it cost virtually nothing to do that, because that's part of the funding for that clinical research unit. The study that I did consisted of the subjects following their amino acid diet for 4 days. They had 2 days at home, then they came in the hospital for 2 days on the amino acid diet. Then we switched them over to the GMP diet, and all those foods for it were made in the CDR, and they were made from GMP that Mark's graduate student, Caitlin, had hand isolated on that Bruker column, okay? So 4 days of food for 11 people, it's quite a bit. And here you see a little bit of data. I won't go into the details of it, but you're essentially looking at the changes 2.5 hours after a meal in total plasma amino acids and blood urea nitrogen. But you can see when you switch over to the GMP diet, the postprandial total plasma amino acids go up and blood urea nitrogen goes down. This is consistent with increased protein utilization. Less of the carbon skeletons are being degraded for urea synthesis. Also, insulin levels were higher. So this is just one little idea of how much better GMP is for the physiology compared to an amino acid formula, in addition to the fact that it tastes pretty good. Tastes much better than the amino acid formula, in fact. So this metabolic study, that was the phase 1, this resulted in, I disclosed to WARF, got a patent. The patent was licensed by a family-owned company called Cambrook outside of Boston. This family-owned company was started by a mother. She had 3 kids, 2 had PKU, and she was tired of her kids being hungry, not having any good food to eat. First thing she did with that company was to figure out how to make good tasting low protein bread. Okay. So 2010, the products were brought to market quite successfully. You see them here. And this little company, Cambrook, is the beautiful business tract. They attracted venture capital big time into that company. They needed it in order to launch this new GMP medical foods, and they were acquired not too long ago by Ajinomoto, one of the largest manufacturers of amino acids in the world. So everybody wins. Everybody wins here. Wharf is making royalty money. The PKU community is well served by this, and business community grows. Okay? Now, to move on to our mouse studies that were conducted for the PKU research, I would describe this as Audrey Gerard spoke yesterday, very nice talk about flavins and improving whipped cream. And she talked about a tangent. Okay, well, that's what this was. Okay, we had extensive mouse studies, hundreds of mice, maybe thousands, probably thousands, lots of papers. I studied everything in terms of PKU to understand the effect of this protein on the physiology. Studied the brain, the kidneys, the bone, the gastrointestinal tract in usually factorial designs. So we had the PKU mice, then we had the wild-type littermates. We had both sexes. They were long-term studies. We had unexpected findings, which I will sum up as GMP burns fat and builds bone. And these findings were especially pronounced in the female mice, especially the wild-type female mice. We used metabolic phenotyping to determine food and water intake and energy expenditure based on oxygen consumption and CO2 production. Now, after listening to all the dairy cow welfare presentations today, they got video all over those dairy barns. It's really fascinating. So this is the mouse equivalent of it, you might say, small scale. We had lots of positive findings in mice, both wild type and PKU, in mice fed GMP, particularly the female mice, and especially compared to casein. And that's important because the casein is the standard optimal diet for mouse growth and maintenance. Okay. And here we show some of the mouse data. If you look in the upper left, we show that in mice fed GMP, we have increased fat oxidation. You see that pink line going across the 24-hour period? That represents the respiratory exchange ratio, the ratio of CO2 to O2. And when that ratio drops, you're burning fat. Okay? And so that is a systemic burning of fat in the mouse. Right below that figure, we look at body composition, and again the pink line, and we see that the mice fed GMP had greater fat-free or lean body mass than those fed casein. They had more muscle, less fat. And bone is in the fat, the lean compartment, okay? Now, this is based on DEXA analysis. We have little mouse DEXA scanners. And then on your far right, we see that the mice fed GMP compared to casein had greater total bone mineral content, and their femurs were longer, and they had a bigger cross-sectional area. Bigger, better calcified bones. You take this together, and particularly in the wild-type female mice. GMP burns fat, promotes muscle, and builds bone in mice. Now, the glycosylation of GMP with the prebiotic properties provides for fermentation of the poorly digested carbohydrate into the production of short-chain fatty acids. And short-chain fatty acids are very positive for health, okay? They will increase gut barrier function. They have been shown to decrease inflammation at both the local level in the gut as well as systemically. And there's also evidence that they promote mineral absorption. Zinc has been documented, and there's also evidence for calcium. Now, this increased short-chain fatty acids with prebiotic GMP has been shown in my lab. It's been shown in other labs in rodents, and there's also human data cited there to support it. And inflammation, and again, the use talk from Brad Bowling's lab this morning was really good on talking about inflammation. But inflammation is associated with obesity, and it's one of the mechanisms of all the negative effects of obesity in terms of on cardiovascular health, in terms of diabetes, insulin resistance. But inflammation is also very bad for bone, okay? It reduces bone turnover and bone buildup, okay? Now, in the mice, we characterize the gut microbiota. And you see there, I want to draw attention to the little green slices in the pie charts. And those are desulfovibrio bacteria. They decrease from 35% to 7% with the GMP, and these were in PKU mice. Well, desulfovibrio bacteria are sulfate-reducing bacteria, and they are associated with spikes in the disease colitis, okay? Now, in fact, there's a little bit of human data about GMP in colitis. So Arla Foods of Denmark funded this study. Arla Foods is the largest producer, seller of GMP in the world. They actually put in their pilot plant, which was eventually upscaled because of our PKU research. Okay. But they were looking for other markets for GMP. There are only two companies that sell GMP. Arla of Denmark, which is the big one, and AgroPer, which is a Canadian company with offices in Minneapolis and also Wisconsin. I'm sure you're familiar with AgroPer. But in this study, they had 24 individuals who had an exacerbation. That is, they had active, and it was colitis affecting their distal intestine. They divided these 24 individuals in two groups, and one of them just continued with drug therapy. Then the other group had drug therapy, but they gave them a supplement, 30 grams a day of GMP. It was just a powder that could be made into a shake, 30 grams a day. This went on for a month. And in this pilot study, what they basically learned was that taking this 30 grams a day of GMP was equivalent to having to increase your drug dosage of mesalamine, was the drug they were used with, having to increase your drug dosage to reduce the symptoms of your colitis. Okay, this is a very positive study, very consistent with GMP reducing inflammation. So to summarize the mouse studies, GMP burns fat and builds bone, and it's a prebiotic with positive effects on the gut microbiota. Okay, now we come to our current study, the GMP hunger study, which is a pilot study. Well, what is a pilot study? Okay. In this case, the goal of this study is to generate sufficient data for Karen Hansen, my collaborator, an MD collaborator, and I, to resubmit our NIH grant to secure sufficient funding, probably at least $1 million, because that's what it takes to do a human study, to secure the funding so that we can do a comprehensive study of the effects of GMP as an aid to weight reduction and positive benefits on bone health. So there's Karen, my friend and long-term collaborator. Karen is a professor in the School of Medicine here, and she's a rheumatologist by training, but her research expertise is bone metabolism. Now, we all know obesity is a huge problem in the United States and in the developed world. And we clearly need some new nutritional approaches to address this problem. And it's a bigger problem in women than men. And women also have the additional challenge, particularly in the postmenopausal years, of osteoporosis. So to put this in scale, so to speak, with PKU, there are 50 to 60,000 people with PKU, treated PKU in the developed world. Now we're talking about 50 million women just in the United States who have obesity. So we're sort of upping our game here, to say the least. The thing that is more recently appreciated is that when you have obesity, you likely have some detriments in your bone health. Used to be thought that, okay, we have more mass, weight, putting stress on the bone, stimulating turnover. And now we know that women who are obese actually have reduced bone mineral density and some deficits in the architecture of the bone contributing to higher risk for osteoporosis. And what's also important to realize is that when you lose weight, which needs to be, lost to promote overall health. But when you lose weight, you also lose bone. So you're putting yourself at a little bit more risk if you're an overweight postmenopausal woman. So we think we have a twofer here. Help you lose weight, but we're going to help you hold on to the bone. It's well known that protein is the most satiating nutrient, more than carbohydrate or fat. And in fact, the evidence would suggest that a high protein, low carbohydrate diet has the most efficacy for weight loss. But now there's an interest in looking at in specific different types of fat. And there's a lot of evidence in both animal models and humans that whey protein has pretty significant anti-obesity effects. This is a study conducted by Nestle in their research unit in Switzerland. And on the left box, you can see they compared energy expenditure after a test meal, and they observed that the greatest energy expenditure occurred after a test meal with whey compared to casein, soy, or carbohydrate. This is an example of some of the strong data supporting the anti-obesity effects of whey. So whey is a pretty complicated mixture of proteins, whey protein isolate, whey protein concentrate. I think there's at least 15 proteins in there. And GMP is abundant in whey. It's in the top 3 proteins. It comprises about 20% of the protein in whey. And we would posit that GMP is a key ingredient contributing to the anti-obesity effects which have been assessed using whey protein concentrate, the mixture. There is evidence in both the rodent and the human literature that GMP promotes satiety, the changes in gut hormones, and also changes in postprandial levels of branched-chain amino acids. This is a mixed literature. It's not all positive. Some of it shows no effect, but it's got to do with how they did the study, how much GMP they gave them, and what did they compare it to. Okay. Now on the right, you see some of our data, and you see levels of ghrelin after eating a GMP breakfast. In the far right, you can see 2.5 hours after breakfast, ghrelin levels decrease. But after an amino acid breakfast, ghrelin levels don't go down. Of course, these are in PKU subjects. Nobody else would take an amino acid diet. And so we don't see that decrease. Ghrelin is called the hunger hormone, okay? It's made by the stomach, and it signals through your brain that you're hungry. When you get up in the morning, you're fasted, your ghrelin levels are high. I think mine, I'm starting to think about dinner. I think mine are probably a little high now, and I'm working up here, okay? But then after you're eating, it's supposed to go down, okay? And that signals you're full. You stop eating. Well, we saw here that it didn't go down compared to GMP. Veldhorst in the Netherlands has the same experiment in healthy, normal people, looking at ghrelin levels. Okay, so this is one of the satiety hormones targeted to assess the satiating effects of GMP. So our objective is to evaluate the effects of a GMP protein supplement. It's a powder mixed in water. We're looking at 2 doses on satiety hormones, food intake and body weight, indices of inflammation, glucose, homeostasis, and composition of the gut microbiota. We are studying 10 women. Why are we studying women? Because they're the ones with the biggest problem. They also are more likely to buy a supplement than men are, okay? From a, long-term view of this. We're going to study 10 women with our DIH grant. They need to be within 10 years of menopause, between approximately 50 and 65 years of age, overweight but not too heavy, BMI of 28 to 35. The protocol takes about a month. It involves 4 visits to the clinical research unit, where we will collect urine and stool fecal samples. The nurses will put in an indwelling catheter, and we will obtain 8 blood draws after eating a test meal containing either a soy control substance or GMP. The GMP supplement that we are using was developed by AgroPer, free of charge. It's delicious. I have 2 flavors, vanilla and chocolate. It's really good. Mix it with water. Okay, it's better if it's cold. Shake it up. There you go. So it provides 130 calories and 25 grams of protein. Now, the low dose of GMP consists of taking the supplement for 7 days. You take it before breakfast, you take it before dinner, okay, and that's for a week. And then the high dose consists of taking the supplement 3 times a day before each meal. We think it'll fill people up and they will not eat as much solid food after taking the supplement. I have enrolled 3 subjects. 1 subject had her visit this morning, which I couldn't go to, but we took care of her. But last week I did visit one of the subjects, and this individual was quite positive. She just finished the low dose, 7 days, and she said, oh, I like it. She said, I've tried a lot of things. She said, I've been on Weight Watchers. She said, I've tried a lot of protein supplements. She said, this one is better. You need to bring it to market. She said, it feels good in my gut. I don't have gas. I don't have bloating. I don't have heartburn. She's very positive about it. That's my personal experience with it also. I think AgroPer did a very fine job. So the hypothesis is we think that GMP will curb hunger and reduce inflammation. The mechanisms include changes in gut hormones, ghrelin, and also cholecystokinin. Also, changes in the gut microbiota leading to reduced inflammation that we believe will support bone health and also probably improve calcium absorption, although that will not be measured in this study. That's for hopefully the NIH study that it will get funded. And to conclude, I've told you one story about GMP, how it can be used for personalized nutrition, which clearly changes people's lives. The GMP medical food story is absolute home run. And I can't tell you how satisfying, Mark, it has been for us to work on this very interdisciplinary project for which I had so much support across this campus. Actually, when I think about it now, I'm not sure it could have been done any other place because we have all the pieces here. We have a CDR. We have a food scientist / chemical engineer, Mark. I had me that I knew the biochemistry and how to do these human studies as well as the animals. We have the Weizmann Center. We have the population. We have the clinical research unit at University Hospital, which made all of this top-notch in terms of doing these phase 1 and phase 2 studies. So yeah, those of you who are new hires, there's a lot we can do if we work with each other, if we collaborate and communicate. And let's hope we have a good outcome for the protein supplement for women to help promote weight loss and preserve bone health. Now, I still need 7 good women for my study. So, and it turns out most of my friends are too old or too thin. I've been through a lot of them already. So if you are interested, call Karen Hansen. The phone number is on the poster. You would be eligible if you're, let's say, about 50 to 65, no health problems, and you have to be a little heavy. It is not an onerous experiment at all, a protocol to be involved with. You'll learn a lot, I think, about your health. And when you complete the study, you get a check for $825. That's part of my grant expenses. So thank you very, very much for your attention at the end of the day, and I'd be happy to address any questions.
Denise, this is kind of a basic question, I think, but phenylalanine is an essential amino acid, correct?
Correct.
So then I would assume that you're seeing, or that people with PKU are seeing some limiting amino acid effects for like muscle development and all that?
Well, see, that's the challenge of the diet. You cannot eliminate Phe from the diet. The kids wouldn't grow. So you have to give them just the right amount of Phe to promote the life cycle growth. And of course, that's going to change all over the life cycle. Within the diet, you do that by the fruits and vegetables. You see? Yep. And then you make up all the other amino acids in the medical food. Okay. So it's a little tricky, and it changes throughout the life cycle. The highest degree of complexity for the nutritional management is when a woman with PKU becomes pregnant. Because even though that baby probably doesn't have PKU, if her blood Phe levels are high, she's going to expose the baby she's carrying to high Phe, and it will interfere with their brain development. Very, very serious. So it's a very complicated management that it's really delegated to the dietitians with a specialty in working in this area. And it's just, there are many, many disorders like this. PKU is one that we know the most about. Now, but you bring up something else, that phenylalanine is an essential amino acid. But remember, if you have PKU, you can't make tyrosine. Tyrosine is not an essential amino acid for any of us, but it is for PKU. So that's a problem because tyrosine, well, that's not very soluble, is it? Aromatic amino acid. So that's going to come out of solution in the amino acid formula. So they have a saying if you're on amino acid formula. and that is shake it up and drink the sludge, because that's the tyrosine. But with the GMP formulations, it goes into solution a lot better, the tyrosine. That is another fine point of the nutritional management.
Hi, Denise.
Sure, Heidi.
You see a lot of people now embracing different kinds of supplements in their diets, regardless of what research has been done. I don't know if you've looked into this, but are there people who are taking GMP for the kinds of reasons you're hoping to study without that research backing of proof?
I don't think so, because it's not sold straight
Okay,
out.
so you
as-
can't buy
as
it.
GMP. You could, yeah. I mean, it's kind of expensive compared to like whey protein because of the extra processing to isolate it, you know. But GMP is very, very interesting bioactive peptide, and it has some antimicrobial effects. There's a patent on it in terms of dental decay. They've shown that it is for the microbes associated with dental decay. It's particularly effective. So it's in toothpaste. You know, and actually I'm beginning to think now, if I'm, you know, my people who are drinking a GMP supplement, maybe it'll help their teeth. I don't know. You know, but Mark, do you have any other thoughts about it? It's a generally recognized as safe ingredient.
That, you know, it's a little bit more expensive than just whey protein, but it's not horribly expensive. It's not expensive like a drug or things like that. And I think that there's no reason it couldn't be made available to the public. It's just that, you know, I think that after your study, hopefully it'll be in every CVS and Walgreens. But, you know, I think right now it's just off people's radar is the main problem.
Yeah.
And so until there's a demand, then there's not going to be a supply for it.
Yeah. I think there's really an opportunity to make a targeted supplement for people with inflammatory bowel disease, you know, colitis and Crohn's. And I've tried to facilitate that and talk to some industry people. There really is not such a supplement. Nestle used to have a supplement that was made from casein, and nothing like the evidence in terms of the anti-inflammatory effects that this small pilot study showed.
Yeah, I had one other comment. You said something about how this work, it would be very hard to see this work have been done at any other university, and I believe that's true for the reasons you mentioned, the Center for Dairy Research and the Center for Dairy Research and the Weizmann Center, the hospital, you know, and all the people that are on campus that you engaged in this study. But I think the other part of it is what you did. Because what you took is kind of like an artist, and they have their color palette, and they're dipping their brush in all the different colors and making a picture from it. That's what you did, as you brought all these people together for a purpose. And I think that, you know, that is a good model for other people to do something significant, is not to go it alone, but how do you put together a team and engage the right people in a team to do something that's bigger than any one person on the team?
Yeah. Thank you, Mark. You know, I have to tell you, I was passionate about it. And part of my passion was that I started my career as a registered dietitian, and I worked in this area of genetic diseases. I worked in the ultra-rare diagnostics. I never saw anybody with PKU. You know, it was much more, but I understood the problem, you know, and when the opportunity was presented me, when I was asked to help, I had a vision. I saw it immediately that we needed to replace this amino acid formula so these children, and adults, so they could have enough to eat and follow their diet. Because it wasn't working. It was the best we had, but it wasn't. So I saw it, and I was more senior. I had been department chair for a number of years, So I wasn't afraid of collaboration and interdisciplinary work. And the other thing is I sort of had the attitude, because I had this passion, that, well, if I didn't know how to do it, I was going to find somebody here that did. And when I asked for help, I always got it. And that was sort of different than like being department chair, because then everybody, I was trying to get people to do things they didn't want to do when I was department chair. But this, it was like, oh, that's interesting. I could do this, and everybody came through, But, and Mark, you had a passion for it too. I think about the story that you told about, remember when you wrote the article for the PKU News about this, and share that with the group. And
for me about this project is I had been doing research for 20 years and publishing papers, and I never got a mother calling me up saying, thank God for your work. My children, I go to bed at night worrying about my children being hungry. I pray for your work and that your work is successful. And I started getting emails like that from mothers and families. And it's very inspiring to get that. So it was, that hooked me.
yeah, exactly. That's the passion. I had it from my history, and then when you had the contact with, then it's the passion. Let's do something that makes a difference here. And never thought about making money or whatever, but we did, So it was the passion that drove it. And we never thought about making money or, whatever. But as Mark taught me, the quickest way to get it to people is commercialization. It's true. That's what WARF does, And sure.
I have a quick question. First, out of curiosity, why did you choose soy as a control protein that is not a main protein in Western diet? And related with that, if you think that some of the benefits are related with the high content of branched-chain amino acid you mentioned in GMP, especially in postmenopausal women that are also having muscle loss.
Okay. Boy, those are good questions. So the reason we use soy as a control for the current study, the GMP hunger study, is because soy is a rapidly digested protein, as is GMP. So then you are comparing a similar entry of amino acids into the plasma after a meal. Many people use casein because casein is more common. And I told you there were conflicting data in the GMP literature, a lot of it because they're using casein. Casein's completely different. Casein forms this big curd in your stomach. It's hard to digest, and it's what they call a slow protein. The amino acids are absorbed quite slowly after a food containing casein. So then we get to the branched-chain amino acids. So this is actually very controversial right now. I don't know, on this campus we have Dudley Lemming, and he purports, and it's supported by some good evidence, that high branched-chain amino acids promote metabolic syndrome and some difficulty in metabolism. Okay? And here I'm feeding high branched-chain amino acids because it's about the plasma amino acid profile. It's, they're up postprandial like 30-40% compared to the soy. And this requires further investigation. There's no question about that. The branched-chain amino acid literature is purporting that could be harmful. It's all based on mouse studies because you can't eat food that has low branched-chain amino acids. Branched-chain amino acids are in all foods, and so there's no human data to support this hypothesis. We do know that branched-chain amino acids constitute, almost 25% of body protein. They're more highly represented in our body's tissue and protein reserves. So the requirements are high, and of course we know that branched-chain amino acids are special. They're the only ones that can be catabolized first pass in the muscle. Leucine is your most important one, a signaling molecule besides a building block of protein, turns on mTOR, protein synthesis. So there's a whole other literature that if you want to build body protein and maintain it, branched-chain amino acids are really good and support that. So we're going to have to shake this out. And I'll be able to, because we're looking at glucose homeostasis. I've got 8 blood draws. I've got insulin, glucagon, I've got glucose levels, I can calculate HOMAs, everything in the study that I'm doing now, so that I will be able to say something about glucose homeostasis. Now, just as an aside, I said that Karen and we submitted an NIH grant that wasn't funded, but we got a good review. And one of the things that they asked us to do was to look at glucose homeostasis. We didn't have that in there. We're all bone and muscle, you know. So that's why the study that we're doing right now, and it was a good suggestion. I think there's something there. But thank you for your very insightful questions.
Thank you. And can we thank Denise one more time? Thanks, Denise, for ending us on such a great note. So thank you once again, everyone, for coming to our first Dairy Symposium. We'll be doing this again. When you get your email as a follow-up with the survey, please let us know what parts of it you liked the most, what things you'd love to see in the future. We welcome all the feedback. For those of you who had posters, you're welcome to take your posters with you, hang them in your halls so that everybody can see the work. The tubes are next to your poster boards, and you're able to take those with you as well. So thank you. We hope you stay connected with us and watch for more updates as the year goes on. And we appreciate your time, hopefully yesterday and today, and look forward to seeing you again. Thank you.